First of all, I would like to say a very good evening to all the panelists and also to our beloved attendees who are attending this webinar 2020 series, which have been organized by JIS Institute of Advanced Studies and Research, Kolkata. Thank you very much to all of you for joining this particular webinar. Before I am going to introduce our speaker, today's speaker, Dr. Dibyendu Kumar Sasmal, I am taking few times to say a few words about our institute very briefly. Basically, JIS Institute of Advanced Studies and Research, in short, JISIASR, Kolkata, is a newborn institute and a little less than a years old. So I just want to share one of my screen. Then it will be good for the attendees to this is the full name of our institute, and in short it is JISIASR Kolkata. And we aim to drive translational research oriented education at the postgraduate and PhD research level in the area of data science, health science, and interdisciplinary science. We are offering different MTech and MSc course in the umbrella of this institute. And for example, you can see here, basically we are, at the moment, we are offering different MTech and MSc programs. And for example, MTech in computer science and engineering with specialization in machine learning and also specialization in data science. Apart from that, we are also offering courses like MTech in bioinformatics and MTech in sensor technology and IoT, along with MSc programs in medical biotechnology and bioinformatics and MSc in polymer science with specialization in paints and coatings. As I mentioned, this is a newborn institute, so we are doing a lot of activities in research and particular academic domains. So I request our attendees please go for our Facebook page to get connected with our present things, what we are doing at the moment, and also please visit our website www.jisiasr.org to know more about our institute. So, okay, I am now coming to today's talk for this session. It is my pleasure to introduce our speaker, Dr. Dibyendu Kumar Sasmal, who has a lot of experiences in the fields of protein structure, function, and dynamics. Other than that, his biophysics and spectroscopy, as well as T-cell immunology and neuroscience, all those fields belongs to his research domain. And he is particularly expert in various kind of technique. And I would like to also mention about his expertization in the techniques like single molecule imaging and fluorescence spectroscopy. And very briefly, I would also like to mention about his, background. When I gone through his CV, I found that he did his PhD from Indian Association for Cultivation of Science, that is called ISCS, situated in Kolkata, and followed by he did several postdocs at various famous institutes such as Bowling Green State University in USA, after that University of Chicago and Argonne National Lab, all situated in USA, and presently he is an assistant professor at Indian Institute of Technology, Jodhpur. He has published several high-impact journals in his research fields. Anyway, today's webinar, what he is going to present here, that webinar will focus on the application of single molecule process imaging technique for studying function and signaling of T-cell. So, without not making much delay, now I would like to request Dr. Sasmal to start his talk and continue his presentation. Dr. Sasmal, can you hear me?
Yes, I can hear you.
So first I would like to stop my screen sharing, and now you can start your screen sharing, and then you can start. While you are doing screen sharing, at that time I would also, for the attendees, I have a special request. If you have any questions and comments, please write it at the question and answer section at the bottom of your windows, and our speaker will address all those after finishing his talk. Thank you. So, Dr. Sasmal, it is all over to you, and you can continue. Thank you.
Can you see my screen now?
Yes, it's fine, absolutely fine. I hope all the attendees also there can also have no problem.
Okay.
Right.
Good afternoon, everyone. Thanks for joining my webinar presentation today. I'd like to thank Dr. Subhankar Singh for his kind invitation for this webinar presentation and his nice introduction. Your institute is very promising. I hope this will grow much faster. I hope all the participants here joining in this webinar are healthy and safe in this pandemic situation. Thousands are dying because of this disease, and scientists are trying to make a vaccine to stop this pandemic. In the meantime, scientists are also talking about boosting your immunity system. So when we are talking about boosting immunity system, a subject in biology comes here, which is known as immunology, and specifically T-cell immunology. So today, I will talk about how in immune science, a very basic thing in immune science is T-cell. It distinguishes between good cell and bad cell. So today, I'll talk about single molecule imaging and its application to study T-cell receptor activation and signaling. So I see there are a lot of participants from all over the India, as well as Korea, US, Philippines, and many other countries. So I decided to start with very basic of single molecule imaging. And here's my laser pointer. If you see this red color dot, that means this is a pointer. I'm trying to get your attention here. So in single molecule science, the question is how to see a single molecule. There is a limitation which is defined by Bragg's diffraction limit. Bragg's diffraction limit says that you cannot image beyond lambda by 2Na. That's the diffraction limit. Lambda is the wavelength of light you are using for imaging, and numerical aperture is equals to n sine theta. Single molecule size is usually 1 nanometer. So the question is, with a resolution of 200 nanometer, if you use a shortest wavelength of visible light, which is 400 nanometer. So if you put 400 nanometer here, the diffraction limit would be 200 nanometer. The question is, how can you use diffraction limited imaging technique to see a single molecule which have a size of 1 nanometer? So scientists came with very simple and innovative idea that if you have to maintain two conditions, the number one condition is you have to use very dilute solution of nanomolar concentration, and your observation volume should be restricted in 1 femtoliter so that you have only one molecule in your focal volume. Now, if you do little bit calculation, the focal volume is calculated as lambda cube, which is equals to 10 to the power minus 15 liter is equals to 1 femtoliter. Now, if you use 1 nanomolar solution, the number of molecule in that solution would be 6 into 10 to the power 14 molecule per liter. Now, number of molecule in focal spot, which is 1 femtoliter, if you calculate, that would be 0.6. Now, the question is why this number is fraction? The reason is that the molecule is always diffusing. So it's diffusing in and diffusing out, and 0.6 is actually an average number. Now the question is why single molecule is so important? It can investigate structure, dynamics, and function of individual molecule at real time. And then it also removes the averaging averaging cross of population. So the data you can get because of average, you can actually remove that. It also explore the hidden heterogeneity in your molecular structure. Let's say you have a molecule with a naive state, also denatured state. So you can actually explore the heterogeneity, how much percentage of molecule remain in native state, how much percentage of molecule remain in denatured state. You can also study directly the dynamics interaction of individual molecule at a time. So single molecule science is essentially a interdisciplinary science. It not only come from chemistry, not only you can apply to physics, not only you can apply to biology, you can also apply biotechnology, material sciences. It also have a lot of instrumentation part, data analysis, computer analysis part. So it very much a interdisciplinary science comprised of a lot of other fields, engineering science as well. So the experimental setup for this single molecule imaging, you need to use the first, you need a laser light, which may be CW laser or pulse laser. It goes through different type of lens, and then it is reflected by a special type of mirror, which is known as dichroic mirror. So this dichroic mirror is a special type of mirror which reflects one color of light and transmit another color of light through it. So this laser light is reflected by this dichroic mirror, and it goes to the sample through an objective with high magnification, maybe 60 to 100x magnification, as well as very high numerical aperture, maybe 1.2 to 1.45, something like that. And then fluorescence from the sample comes back through the objective and transmits through the dichroic mirror, and it blocks laser light, any laser light, with this emission filter, and then goes to a confocal pinhole through the lens. A pinhole is important for a confocal microscope because this pinhole of size of 50 micron, maybe 20 micron, 20 to 50 micron, it actually blocks light, anything coming out of the focus point. So any light or any signal coming from above the focal volume or below the focal volume is blocked by this pinhole. So this is how you can actually increase signal to noise ratio, which is important for single molecule imaging. And then you have a very important component of this whole system is called detector. You can use either avalanche photodiode or electron multiplying charge coupled device known as EMCCD camera. So the price, the price of this camera is actually very expensive, which is equivalent to a BMW, almost 30 to 40 lakhs in Indian rupees. So if you look at single molecule imaging, here every dot is a single molecule. Now you can ask me that every dot could be a multiple molecule, right? This is the right question. But there is a way to resolve whether there is a multiple molecule within that dot or not. You know, the other way to see single molecule is that if you see a kind of fluctuation of fluorescence, something like this, that means there is a single molecule, and then each peak of this fluorescence is because a molecule is diffusing in and out of this this focal volume. So this is why you have a fluctuation of fluorescence intensity. Now, there are many single molecule technique. My group is expert in a single molecule technique known as single molecule fluorescence resonance energy transfer. In short, it is known as SMFRET. This is also known as spectroscopic ruler because you can calculate a molecular distance in between 1 nanometer to 10 nanometer in real time. So this is the beauty of this technique. In short, the principle of single molecule FRET, many of you may know, but I found there are many students and postdocs are joining in this webinar. So briefly, if you excite a donor with a light, it will go to excited state. And then after a solvent relaxation, it can either give a fluorescent signal or it can transfer the energy to a neighboring molecule which is within 1 to 10 nanometer, so that the other molecule, which is known as acceptor, will accept that energy, go to the excited state, and then it will give a signal for acceptor. So the thing is that if you attach a protein molecule with a molecule donor and attach another molecule acceptor, then you can actually calculate the distance between donor and acceptor in real time, so that you can also know the heterogeneity of the structure of protein molecule. The efficiency of freight is known as, is also be calculated like this, intensity of acceptor divided by intensity of acceptor plus intensity of donor. I have published a review paper in nanoscale. You can actually read that paper. I have extensively described the principle of fluorescence resonant energy transfer and its application and its limitation and many other things. So you have to meet two conditions for a successful freight experiment. The distance between donor and acceptor should be within 10 nanometer, as I said before. So if donor and acceptor, they are close to each other, then energy from donor would be transferred. So you have an intensity of acceptor high. And then if donor and acceptor distance is longer, then energy transfer efficiency is low, right? So you have an intensity of donor longer, higher, or intensity of acceptor smaller. Another important parameter for a successful freight experiment is that the overlap integral between emission of donor and absorption of acceptor, there should be a significant overlap between these two curves. Okay, so I'd like to explain single molecule freight, how you can actually apply to study dynamics. You know, let's say there's a butterfly and you labeled a donor here and acceptor here, and when the butterfly is actually flying, there will be two types of conformation, either this one or this one, and in between of that, right? So when they are the wings are close to each other, that means energy from donor is transferred to acceptor. So the freight efficiency is high, right? So the rate is high. So here is this conformation. And at the same time, the green color is quenched. So this way, the donor and acceptor signal would be anti-correlated to each other. Now, you can apply the same thing for a protein molecule. So here, as the distance between donor and acceptor is longer, there is a low freight efficiency. So we plot here freight efficiency versus distance between donor and acceptor. But if another conformation, which have a little distance or shorter distance compared to here, the freight efficiency would be much higher. So you have a higher freight efficiency. Now here you can see when donor is quenched here, green color, at the same time acceptor is increased. So this change is because of the fluctuation of conformation here, this one and this one, this one and this one. So it will continue up to 160 seconds. So the beauty of this experiment is that you are actually observing the molecular conformation at real time. At the same time, from this data, you can calculate the efficiency of freight. So when you have a higher acceptor intensity, so at the same time, your freight efficiency is also very high. Now, in context of T cell, I would like to introduce you now this is the main topic of my discussion today. A T cell is a subtype of lymphocyte. It plays a extremely critical role for immune response. A same image of TCL is here, and here we have a red blood cell, platelet, and T cell. There are different type of T cells in blood depending on the function. There are helper T cell, which is also known as CD4 positive T cell, which is activated by peptide major histocompatibility complex type 2 molecule, which actually helps to other cell to get activated. Another type of cell is known as cytotoxic T cell or CD8 positive cell, which is activated by peptide MHC class 1. And this is extremely important cell because it kills cancer cell. And there are memory T cell. Memory T cells also is important because it's an antigen specific and it remains in blood for a longer time after the infection is eliminated. So when a vaccine will be discovered for this COVID-19 pandemic, so what will happen? The vaccine will be given to the patients, and then after that small infection, the memory T cell would be generated in the blood, and so this will be always remain in your body, and it will protect any other infection which may come later. That's how our immune system protect from any disease. There are other type of T cell is known as regulatory T cell. It actually shut down any T cell mediated reaction. There are many other T cells, NK cell, natural killer cell, gamma delta cell, alpha beta cell. So I'm not going to mention all of them here. But if you look at T cell activation pathway, so what happens when a B cell represents a peptide histocompatibility complex on the surface, then T cell have a T cell receptor on its surface. So peptide histocompatibility complex and TCR binds together, then what will happen? T cell will be activated, and then T cell will be converted to many other type of T cell, or it can actually generate a chemical messenger molecule which is toxic, known as cytokine. It can also kill other type of antigen presenting cell. So when T cell and antigen presenting cell form a bond, the area in between T cell and antigen presenting cell is known as immunological synapse. I want to show you a real video which we acquired here. See how T cell actually killing 2 cancer cells. This is known as apoptosis. So all happens because T cell bound with peptide MHC, T cell bound with peptide MHC, and then T cell get activated, and then it release cytokine so that the T cell, so that cancer cell, these 2 cancer cells become, they get killed because of that cytokines. Okay, so if you look at that interface in molecular level, there are different type of receptor molecule on the surface of T cell, as well as there are different type of ligand molecule on antigen presenting cell. The most crucial molecule here is TCR on the surface of T cell, and then peptide major histocompatibility complex class 1 or class 2 on the surface of antigen presenting cell. And another type of molecule here is immune checkpoint molecule, which is PD1 and PDL1. Now, if PD1 and PDL1 interact to each other, so there will be no signal. That means T cell will not be activated. Very recently, there is a Nobel Prize of 2 or 3 scientists, I forgot. They got Nobel Prize because they discovered PD1, and that's how immune therapy is actually discovered. So my focus on this talk is how this TCR and peptide MHC interact each other, how this TCR discriminate different type of antigen. So if you look at more detailed TCR peptide MHC complex, here we have 5 different type of there's subtype alpha, beta, and then gamma, delta, epsilon, and then zeta chain also. There's a co-receptor molecule, CD4, and then we have a peptide MHC class 2 molecule here. Okay, so when we started this project, just before that, there was a paper in Immunity in 2013 that specifically showed that even if a single peptide MHC molecule can trigger a whole activation of T cell. And this is important. Here you can see with time here up to 120 minutes, the number of T cell receptor molecule actually increase with time. That means is single peptide MHC can also generate a full scale of T cell activation and signaling. Now, all the background here, that there's a different type of model available to explain how TCR-pMHC is activated, how TCR discriminate different type of peptide MHC. But the only problem of those kind of study is that there is a lack of a definite technique which can directly visualize the TCR and peptide MHC interaction. So we have a state-of-the-art technique which is very, very advanced, single molecule Fred imaging. So we thought, why not we take advantage of that technique to apply and study how TCR discriminate different type of peptide MHC molecule or antigen molecule. So our hypothesis was TCR undergoes a conformational change upon ligand engagement to initiate T cell signaling. So to do that, we developed a very advanced microscope in our lab. So what we did, we have a 4 different color of light, a laser light. These are all CW laser light. And then we also have an analog multicolor LED light. And they are all focused to the objective, to the sample. Now we have a magnetic objective. Here we have a 100x 1.4 NA magnetic stage and 100x 1.4 NA objective. This is a TARF objective. And then fluorescence from a sample goes back through that objective and focused again side by side on an EMCC. The beauty of this, the whole microscope system, is that it can measure a 6D, which is X, Y, Z, wavelength, time, and multi-point. And the application is enormous. DIC from FRET to polarization FRET to FRAP to epifluorescence, TARF, STORM, super resolution microscope, and time-lapse microscope. So this is the microscope, real image of microscope we built here. We have 4 different type of laser light from cobalt, and they are all laser are focused in such a way that all light will be focused on this optical fiber. And this optical fiber is connected with the TARF arm. So here is the microscope setup, and this is the EMCCD I told you. The price of this one is similar to BMW. Well, so to solve that problem, what we planned, we'll build a glass supported lipid bilayer on glass surface. And then lipid bilayer will be labeled with peptide histocompatibility complex, and then we'll add a T cell. That T cell would be labeled with a single chain antibody fragment. This is a fragment of an antibody, SCFV, and which is labeled with a Psi5 dye here. And then we have Psi3 on peptide side, so that we can do a FRET experiment, single molecule FRET experiment. As I said, single molecule FRET experiment is a spectroscopic ruler. You can determine distance in between 1 to 10 nanometer in real time. So what we decided, we'll change a peptide sequence from here, from super agonist to weak agonist, and we'll try to understand how T cell actually discriminate this type of peptides or peptide histocompatibility complex using single molecule imaging. So before that, we have to do a couple of standardization, the first one is lipid bilayer. So what we did a FRAP experiment, fluorescence recovery after photo bleaching. So if video starts again, I'll show you here is a black spot. That black spot is because of the photo bleaching. See, the high power laser is focused, and then photo bleaching, and because of the diffusion of that molecule, 2D diffusion of peptide MHC molecule, so they actually diffuse to that area. And then you can see how nicely recovered, almost like a 90% intensity is recovered. So this actually says that lipid bilayer, what we planned, it will be used for antigen presenting cell have a nice fluidity and integrity. Okay, and then more interesting part comes here. A single molecule tracking of peptide MSC on lipid bilayer. So here, see, each bright spot is actually a single peptide MSC molecule on lipid bilayer. They are diffusing because lipid is in liquid state, and lipid have a 2D motion. And then we did a data analysis. So we started to track each and every molecule so that we can determine the diffusion coefficient of those molecules. So it will help us to determine that lipid bilayer is exactly what we need. And then we also started to track each and every single molecule. So for example, I'm showing here, see, we are tracking each and every step of this molecule. And then finally, we will determine the diffusion coefficient and how that peptide MSC is interacting with each other. It is extremely important to determine that each and every peptide MSC molecule is in single molecule level. Now, there are a couple of ways to determine that, whether that bright spot, each bright spot here is a single molecule. The number one way is you just take a bright spot, and then what we did, we fitted that bright spot with a 2D Gaussian function. You can see here FWHM of this 2D Gaussian function is in X 390 and in Y 385, which is almost similar like a base diffraction limit. The other way to make sure that each and every peptide MSC molecule are single molecule by single molecule photobleaching. You can see here, so molecule is going like this way, and then suddenly it become bleached, and then we have a background. So it basically says that bright spot actually with a single peptide MSC molecule. So these are just 3 different examples. And then we also need to make sure that T cell receptor on the surface of T cell, they are also nicely working and then nicely tracking towards the center of synapse. So see here, we have added just T cell on that lipid bilayer, and then see how nicely each T cell receptor microcluster. So each spot, bright spot, is actually T cell receptor microcluster. So each microcluster may have 100 T cell receptors. So we are tracking each receptor. See how nicely they are actually going outside and then coming inside. It's like a fireworks, right? And the reason is that if you look from here, because our objective is here, right? And then right after T cell activation, the T cell microcluster starts to grow this way. They are on the surface. So they go outside and they come back to the center again. That's why the tracking looks like they're going outside and then again coming back nicely. So this will make sure that T cell receptor also is getting activated by a single peptide MSC molecule. Now, as I said before, we'll do a single molecule Fred experiment with different type of peptide. They have just 1 or 2 amino acid sequence change. Well, here is a single molecule Fred experiment. You can see how nicely single peptide MSC molecule are triggering a single T cell receptor. Here we have a single peptide MSC TCR, and then from that image, we calculated a Fred image here. And then tracking of that each peptide MSC and corresponding TCR will get a nice time trajectory for donor and acceptor. The red one is for acceptor and the green one is for the donor. And then we can calculate the Fred efficiency trajectory as well. So, and from that Fred efficiency trajectory, we got a nice histogram, and from there we can calculate the distance between TCR and peptide MSC bond, which is around maybe 43 angstrom for that specific peptide. So we repeated that experiment and that data analysis. This takes enormous data analysis computation. So we repeated for 20,000 event for K5 15,000 event for MCC and 16,000 event for another peptide, which is weakest agonist 102S. And then what we found very interestingly is that for K5, the distance between TCR and peptide MHC is the shortest, which is around 43 angstrom. For MCC, which is an agonist, the distance between peptide MHC and TCR is around 51 angstrom. Whereas for weakest agonist, the distance between TCR and peptide MHC is around 60 angstrom. In addition of that study, we also did a co-stimulatory receptor CD28 and a immune checkpoint molecule PD1, how they actually remain each other. So during the synapse formation, you can see here, so this axis is 0 second, 30 second, and 154 second. Here you can see how PD1 actually coming to the center of synapse, and then CD20 is coming to the synapse, and TCR also coming to the synapse. Now, if we superimpose all to each other, so you can see when synapse is formed, PD1 and CD20, they are almost co-localized, right? But PD1 and TCR, they remain actually, they don't touch each other. So PD1 remain outside of TCR. So TCR remain inside the center. So this paper was published in Science in 2017. It's a very highly cited paper. Okay, so once we know that how TCR discriminate different type of peptide MHC, the question is how this conformational change translate into the intercellular activation. So to do that, what we planned, we labeled CD3 zeta chain of T cell with GFP, and then we also labeled the antibody fragment with Alexa 568, and then we did exactly the Freight experiment. So here you can see this green color is actually intracellular GFP. So remember when I was showing previous Freight data, there was extracellular Freight, and this is a kind of transmembrane Freight. One donor is inside the cell and acceptor is outside of the cell. So this green color is from GFP. Now you can see how nicely GFP is coming, the CD3 zeta chain actually coming at the center of the synapse as well as TCR. So they are actually co-localized to each other nicely, right? And then from this image, donor and acceptor image, we calculated the Freight efficiency. And then we did a control experiment with glass surface so that the peptide MHC molecule remain immobilized on the glass surface. So you can see here peptide MHC is immobilized. So there is no fluidity. Peptide MHC is not moving. You can see here, although their TCR or peptide, they're doing a little bit cluster, but the synapse is not formed well as we have observed in lipid bilayer. So this basically shows that it's a dynamic phenomena. Synapse formation is a dynamic phenomena, and you must need a TCR and peptide MHC with a fluidity to be activated and to generate the signal. So we did another round of data analysis here, tracking those GFP, as well as we calculated the distance between those two molecules. So here we calculated distance between GFP and Alexa 568, depending on what is the peptide MHC here. So what we found is that for K5, which is the strongest agonist, is actually generate the longest distance between them. That means when TCR is activated by a strong agonist, the CD3 zeta chain dissociate from the cell membrane, and it actually goes right perpendicular to the right on the axis of TCR peptide MHC. So in addition of that, we also interested to know 2D kinetics of TCR and peptide MHC interaction. So we did an innovative way. We took human red blood cell and we attached that with a glass pipette and then labeled that surface with peptide MHC. We also attached a T cell with another glass pipette. So here we have a glass pipette with T cell, and here we have a human red blood cell. attached with another pipette. Now, what will happen if you just touch and pull them apart? Because of the TCR and peptide-MHC interaction, you can see little stretch of red blood cells. See, this one is very nice. Because of TCR and peptide-MHC interaction. So this is the way you can actually directly measure the kinetics of interaction TCR and peptide-MHC. So we increase the ligand density on the human red blood cell, and you can see the adhesion probability also increase. If you increase the adhesion time or contact time, that will also increase your adhesion probability. So with different type of ligand, we can calculate the binding efficiency or binding kinetics. So you can see here for K5 we have 3.6, for MCC we have 2.0, 10 to the power minus 5, and then for 102S there is a almost 1 order difference here. And then we started to think about how this interaction also influence the signaling. So you can see here how calcium is actually released when TCR-peptide-MHC engaged here. It goes through different type of intracellular signaling. One of them is that it releases calcium from ear through this calcium ion channel, and it releases that calcium in cytoplasm. So usually in resting stage, the calcium concentration is 10 to the power minus 7. However, in activated stage, it become 1 order difference is micromolar in activated state. So we thought, why not we actually measure the calcium signaling? So we loaded this T cell with calcium sensing dye, and then here we have, instead of human red blood cell, we have a B cell. And then when they interact each other, and then T cell get activated, and then nicely you can see a color of high calcium flux here. So here red color means that almost calcium is increased by 3 times from the baseline. We did a similar experiment with 3 different dye, with 3 different peptide. We coated that peptide on the surface of this B cell. We have MCC 102S. Also we did a control experiment with a human peptide which do not interact with mouse T cell. So here you can see with time there's no increase. That means this interaction and calcium flux is real. So we can study easily how this calcium is increasing with time, and we can study the kinetics from here. So here is kind of summary in between CD3 zeta dissociation and calcium flux, how they're correlated to each other. So usually in resting stage, CD3 zeta chain, they remain bound to the surface, intracellular surface of T cell. However, once that T cell bound to peptide-MHC, then what happens? Calcium increase, right? I just showed with that experiment. So that actually increased the charge density inside the cell. And then because of that electrostatic interaction, this CD3 zeta chain, it also have 2 positive charge. So it comes inside the cytoplasm. So for any K5, if you use a strongest agonist, this interaction or this dynamics change is actually much faster compared to MCC and 102S. The micropipette system, what we used is absolutely home built. Here we have a micro manipulator, and in right hand side, we usually hold human red blood cell in this micro manipulator. We have another micro manipulator here. We hold T cell. And then we also have 2 water tower here. This water tower is used to generate negative pressure, so that there is a suction so that there is a suction in that position to tightly hold 2 type of cells here. So I'd like to conclude here is that using single molecule freight, we observed directly the interaction between TCR and peptide-MHC. We also measured how T cell receptor discriminate between antigens. So when you have a agonist peptide, the distance is the shortest, but at the same time, it generates intracellular signaling much faster, so that the distance between CD3 zeta chain from TCR is actually highest, around 64 angstrom. In addition of that, if there is a weak agonist, the distance between TCR and peptide MHC is the highest. However, it generates the signaling from TCR to intracellular very slow. That process is very slow. That's why the distance between TCR and CD3 zeta is around 53 angstrom. So the downstream signaling, calcium signaling, activation, affinity, that actually correlated with the conformational change. So this work is already published in a Nature family paper known as Cellular and Molecular Immunology. Part of the work is also published in Science in 2017. And then if you are interested to read or know more about single molecule freight and its application, I wrote a nice review paper. Please go through that paper also. So finally, I'd like to thank IIT Jodhpur, University of Chicago, Argonne National Laboratory, and Bowling Green State University. This project was almost impossible without collaborators. I was fortunate to get a lot of collaborators from different reputed institute. For example, we got Wei Feng from our lab, and Shobhan Roy. He's a nice scientist from Adam's lab. And then Ian Run, she's extremely talented and helped us to generate virus for CD3 zeta GFP generation. We also got Dr. Mike Barbon from MIT. He provided plasmid, peptide exchange protocol. We also got help from Dr. Enfu Hui. He's assistant professor in UCSD. He provided lipid bilayer protocol. We also do a lot of collaboration with Tyrell, Matt Tyrell. He's the director of Institute for Molecular Engineering at the University of Chicago. The whole research was supported by National Institute of Health as well as Chicago Biomedical Consortium, my own funding. I would like to end with a little bit about advertisement that I started a new lab. There's a lot of PhD position open in my group. You can visit my research group at www.sasmallab.org. You can also reach by email. My email ID is sasmal@iitj.ac.in. You can apply this time for PhD position if you have if you are qualified with GATE and NET. The deadline is 30 May. So please feel free to apply, and I'd like to conclude here. Thank you for your attention. It's over to Dr. Singho.
Dr. Sasmal.
Yep.
Thank you very much. But before that, can I request you to stop the screen sharing?
Sure.
Okay. Okay. Thank you very much, Dr. Sasmal, for such a wonderful and insightful talk. And in fact, I was also really amazed. I mean, during your explanation, how you simply explained a very, I mean, somehow little different topic. For example, single molecule imaging. I mean, what you have, I mean, the way how you explained it will be very, I mean, good, or I mean, for our attendees who are from the different field or who are undergraduate students, for them. And at the same time, also later stage, you explained very well how this kind of technique can be used for, I mean, application purpose, especially to know the biological signaling or T cell activation, those kind of biological process monitoring. I mean, right. And your work was really, I mean, very, excellent. I mean, excellent. And but before going to talk about more about, I have few, questions and comment also. But before that, I just need to check whether our attendees, what kind of comments and questions they made. And actually, they are also agree with me. They also mentioned that it was very nice explanation, sir. And a few of them, they also commented that very good and interesting work. And the question what I can ask you that, yeah, there is a few question also. Can you see the question? I should read, because all the attendees, they cannot see the questions, and so I have to
Yes.
read out. So I'm reading the first question, which was asked by Subhajit Chakraborty, and he asked that why is the color change happening from blue to red, shifting of wavelength? Means blue to red is a shifting of wavelength,
So
of course. So in T cell and calcium signaling. So okay, the question I am again reading out completely. The why is the color change happening from blue to red, that is shifting of wavelength in T cell and calcium signaling?
okay, this is a nice question. Thank you for asking this question. The color is actually a false color. So the color changing is just to show that how many times the calcium is increased inside a T cell because of the activation of T cell. So when T cell and peptide MHC, they interact to each other, that interaction translates to the intracellular signaling inside the T cell, right? And that actually generates a lot of intracellular signaling paths. And one of that is that it actually opens up calcium channel from ER, which increase the calcium concentration inside the T cell. I told you that it almost changed like a one order, you know. So there is a 10 to the power minus in resting, 10 to the power minus molar concentration of calcium in resting stage. However, it actually changed to 10 to the power minus 6, which is micromolar. So here, the color is actually a false color. What I'm showing is that the times actually change. The calcium concentration, the times is changed. So if you see if you just play back my video later, you will see there is a color graph, you know, color bar. So the color bar in just right side of the color bar, it shows what color means what time of increase. You know, there is a F by F0. So F is the intensity after activation, and F0 is the intensity before activation. So at the resting state. Now, if you divide that F by F0, that will give you the multiplication of increase of calcium inside the T cell. So this is basically a false color, and the red color means you can actually make any color you want. I just chose a red color for 3 times of increase. You can make even single color. So if you make a single color increase, so what will happen? You're suddenly a single color, it's a green color, and suddenly after that touch between B cell and T cell, the suddenly dim color becomes very bright, right? And you can make the single color much better way to explain that how much it actually increased using a little false color, right? So hopefully I explained you.
Pseudo color imaging.
It's a pseudo color.
Just it signifies that the intensity is increasing.
Right.
Yeah. So anyway, I mean, I also, as you mentioned that, I mean, if the attendees, they want to, I mean, listen this talk again. So for their, I mean, some information, I would like to say that this whole webinar is already live in our JIS Institute of Advanced Studies and Research Facebook page, and that I mean, recording will remain there. So if you want to see or watch this webinar later, I mean, you can also follow that Facebook page, and you can go and watch that one, right? So I mean, as Dr. Sasmal also mentioned, you can again go back there, right? So I found that one, I mean, attendee who was raising hand, but I would request her, the Marli Pakase, I request her, if you have any kind of question or comments, please write on our question and answer section. because in this webinar we are not allowing our attendees to talk because there is a lots of attendees, so it's very difficult if we allow to anybody. So that's why we are asking you, please, if you have any question or comments, please write on the question and answer box or chat box, right? So okay, I'm going to the moving to next question. What is done by Saurabh Sarkar? He asked that which software you use to project the molecular motion in cells.
So
which software you use to project the molecular motion in cell?
So you can use a different type of software for tracking. So what we use for this particular analysis is ImageJ. There is a plugin in ImageJ known as TrackMate. So you can download TrackMate plugin in ImageJ, and you can use that TrackMate. This is extremely useful software for tracking single molecule on cell surface, even in solution phase. So it can actually give the track path each step, how much intensity actually changed, how much distance changed, and it will give also a track path with a neighboring molecule. So it will give each molecule with their own ID. So it's a beautiful, nice and useful software. I highly suggest to use that software. Go to ImageJ and download a plugin called TrackMate.
Okay. Thank you. I hope Saurabh Sarkar, he already got your answer and he got the queries. And then the next question from Devaroti Bag. She is our actually student, from our institute. So she asks that, hello sir, number one question is, can cancer be detected through TCR peptide and MHC interactions? If yes, which type of cancer? And second question is it used only for diagnostic purpose, or this can also be used for therapeutic purpose?
Nice question. Thank you for asking this question. The first question, the answer is no. You cannot detect TCR-pMHC interaction for cancer using this detection. So the reason is that the reason is biology. You have to understand the biology. So the cancer cell is actually very intelligent. That's why it grows. So what happens? It actually generates immune checkpoint molecules. For example, PD-1 or PDL1 on its surface. So that's how T cell distinguishes between a good cell and a bad cell. So when cancer cell grows, it have the same molecule which actually have in our normal cell, like brain cell, tumor cell, heart cell, lung cell, whatever, that normal cell, the same molecule is there. So when T cell comes, that PD-1 and PDL1 interaction happens, right? That immune checkpoint molecule. So that actually gives negative signal to the T cell. So that's why the T cell think actually that cancer cell is actually good cell. And that's why it actually invades, immune system. It actually make fool to the immune system, and cancer actually grows day by day. So you cannot actually do peptide-MHC interaction. So if you want to do real-time peptide-MHC interaction, a single molecule level, I would probably say no. So that is why one of the reasons we use, instead of cancer cell, what we used, we took peptide-MHC molecule on lipid bilayer surface. So that actually simplify, so that actually eliminate that immune checkpoint part, So when you have a lipid bilayer, when you only add a peptide-MHC molecule, antigen molecule, that gives you the only interaction between TCR and peptide-MHC. There are many other molecules on cancer surface, so you want to avoid that interaction at that study. And the second question is this a tool can be used for diagnostic purpose? I believe so. We have a plan for developing a diagnostic tool for that peptide-MHC and TCR interaction by that micropipette system. So if you remember the picture where I was showing that developing a microscope, where you have a T cell and peptide MHC or red blood cell, you know, they come to each other and they attach with time, right? If there is an interaction, then you have a elongation or stretch on the red blood cell. So we have a plan for using that setup for diagnostic purpose.
Okay, thank you. And then comes the next question is from Niladri Sekhar Devroy. And his question is, what is the future scope for single molecule imaging? Will there be any further imaging of other cells which are mainly useful?
Sure. As I said, single molecule spectroscopy or single molecule imaging, the scope is enormous. One scientist, Nobel laureate W.E. Morner from Stanford University, he specifically mentioned that after 2020, every biology paper should have or will have a single molecule imaging data, because a property from ensemble molecule, a property from single molecule is totally different. As we are all different, all human beings are different person. We should not judge, you know, as a group. We should be judged as a single person. So similarly, molecule, they should not be judged as a single, you know, as a group. They should be judged as a single. So they go to the specific point, for example, therapeutic application. They go to that specific point and works as a single. So we need to understand the single molecule, you know, the biology or the scope for that is actually enormous. So one example I want to mention that, so one example is that single molecule DNA sequencing. So which have an enormous application in future, you know.
Okay, now I'm moving to the next question. That one is from Subhajit Chakraborty. And his question is, how close data point can be analyzed in time space, like binding time?
Winning time.
Winning time.
Okay, so it depends on what is your recording, you know, speed. So if you are recording, I know Subhajit is actually trying to understand in terms of fluorescence correlation spectroscopy.
Yes.
So depending on what is your data binning time. So let's say if you have a smallest binning time, maybe 0.1 millisecond or 0.01 millisecond when you record that data. So you can definitely average 2-3 point and make, you can see that 2-3 point as a single point, right? So that's how you can actually increase your binning time when you actually determine a histogram.
Thank you, Dr. Sasmal. And there is another, possibly this is the last question, which is again from Saurabh Sarkar. Actually, previously he asked that the software, which kind of software you use to project the molecular motion in cell, and you already answered that one. And right now he's asking that, can I find it, means that software in Image Pro Plus 6.0 as well? That one,
I have no idea. I have never used this software, Image Pro Plus.
I mean, I would request Saurabh Sarkar, if you really have much interest to know about this software, or I mean image processing, and if you want to, I mean, use that kind of software, so you can directly contact with Dr. Sasmal. He's always available. I mean,
I'd be happy to explore this software if there is any new features.
I mean, I'm also requesting Dr. Saurabh Sarkar to directly contact with you also, right? And then comes another question. Oh, this one is also from Saurabh Sarkar. He asks that, as far as I know, Sci5, Cyanin 5 has photostability issue, right? Is that all right to use it for prolonged time lapse imaging? Is that all right to use it for prolonged time lapse imaging?
Okay, so let me give you this answer first. So photostability doesn't depend only on the issue of dye. It depends on how long you are doing experiment, also the power of the laser you are using or the power of the light you are using. If you use a high power laser, any dye will bleach at faster, you know. So there should be a balance between the power of light, the power of laser, and the photostability of the dye. So I would say, yes, Sci5 have a photostability issue. There is a better dye, Alexa dye. There is a better dye even from better than Alexa, which is Atto. You can also use quantum dot for your experiment, which is much higher, have much higher photostability. So go for that. But always remember, it not only depends on dye property, it depends also the power of light you are using. So there should be a balance between photostability of the dye and the power of light.
Okay, and the next part of his question is the maximum how long such real-time monitoring can be done without harming the cell status?
Yes, so that is why you know, we don't want to kill any cell when you do experiment, you know. That's why for this experiment, what we used a TIRF, total internal reflection fluorescence microscopy, which have around 200 nanometer, you know, the imaging depth in g-axis. So I would say when you do any single molecule imaging experiment with live cell, use TIRF. And if you cannot use TIRF, use at least confocal. And the other way, if you have really good, you know, camera, as I said multiple times, if you have a really good camera, like the price of that camera is like a BMW. So what you can do, you can actually minimize your laser power, but you can actually amplify that signal with minimum light. You can get a minimum signal. However, you can actually amplify that signal so that you can see that cell much better way. So that's how you can actually reduce photo bleaching of cell.
Okay, actually I was thinking that was the last question, but casually so many questions still coming. And next question comes from Manjima Sarkar. And she asks that, good evening, sir. And first she gave you a compliment that, good evening, sir. I really thankful to you for sharing this wonderful session for us. As I am a student of pharmaceutical science, so I would like to asking you that how can we implement this in the pharmaceutical technology and how this helps to enhance the pharma technology. Thank you, sir, in advance. Okay. So I mean, he's asking the application of this kind of technique in pharma technology, right?
Yes. So, so one of the example I want to tell you that we recently got funding. So let me tell you a little bit background how we actually applied single molecule technology for pharma. So there is a disease called muscular dystrophy for kids, Duchenne muscular dystrophy, DMD. So usually, if any, this is a male boy's disease, you know. Usually, if that disease happens to any kind of kids, they would survive 20 years, only 20 years. After that, they will die. So we thought how we can actually implement or apply our technology to develop immune therapy to combat that disease. You know, there are around 20 million kids, and one kid in probably 70 total boys kid they born every day. One in a 70 they born every day with that disease. So they will ultimately die after 20 years. So we thought how we can actually apply this single molecule technology and to determine the early detection, you know, and then how we can actually develop a therapeutic application. So we actually trying to develop a single molecule, you know, small molecule that will be encapsulated inside a lipid vesicle, and that will be transferred to the cell specific affected area so that it can specifically cure that that muscle problem, muscle with problem. So that's how a single molecule technology or imaging can be applied for therapeutic. The number two is, as I said before, DNA single molecule DNA sequencing, which is much faster, much highly efficient. You can You can read their enormous paper on single molecule technology. The other one is basic science research. People are trying to understand how this virus actually go inside the cell. So they are trying to directly observe how virus cell is attached on the normal cell and how it actually deliver the RNA molecule inside that virus to a normal cell. So this is how you can actually use as a diagnostic. Maybe there is a therapeutic application in future.
Okay, thank you, Dr. Sasmal. Then there is another question from Chetan Singh, and his question is this technique is useful in genomic study?
Yes, there is a whole subject called single molecule genomics.
Right.
Yeah. So I'm not an expert of single molecule genomics, but I would refer to go one of the pioneer of this single molecule genomics experiment is Sunny Zia from Harvard University. So I would suggest you to visit his lab website to know more about single molecule genomics.
So I mean, here I just would like to add a few things. That in fact, in our new one institute, JIS Institute of Advanced Studies and Research, we also have a bioinformatics course, and one part of that course, I mean, not only one, several faculties, they are expert in genomics and especially gene sequencing studies, right? So I mean, if any student they have an interest, they can contact with those faculty members also. I mean, regarding that gene sequencing study or genomic study, right? I mean, they can also communicate with this kind of imaging technique. That will be a kind of interdisciplinary research, right?
Right.
So next question I am moving to, that one is from, he did not mention his name, but what is his question is, what is scope for a chemistry student for research in T cell signaling and its application? So
this is a nice question. I would like to tell you that there in higher study or higher research, there is no difference between chemistry, physics, biology, computer science, engineering.
So can I add first before you give the details answer? I'm just reminding all the attendees that Dr. Sasmal, he is from a chemistry background. I mean, he did all his study in chemistry, and later he moved to this field. And here finally he moved to this field, means imaging or spectroscopic field, and later to apply that kind of technique, he moved to biological field. So all these fields are kind of interdisciplinary. So I think Dr. Sasmal, he can explain much more details about his transformation of those kind of things.
I started my research career as a spectroscopist. So we used ultra-fast fluorescence spectroscopy, for example, fluorescence up conversion spectroscopy to study biological water dynamics inside many model systems, for example, micelle vesicles, as well as we developed other microscopy techniques, for example, 2D, two-photon confocal microscopy, that is also time-resolved microscope we developed in Kolkata to study biological water dynamics inside a live cell, the different compartment of the live cell using that confocal microscope. So you can see we are applying a physics technique to solve a biology question, which is actually most interesting thing I have ever seen. So when we apply a physics question, physics tool to solve a biology, that is actually more useful. In today's context, even it is more useful. So people from engineering background, people from material science background, people from mathematical background, they're all trying to understand this COVID-19 pandemic. So is there any difference? This all subject is same, but people are all trying to focus on that problem and trying to solve from different angle. If you're expert from biology, you can do good biology. If you're expert from physics, you can do a lot of biophysics. If you're expert in mathematics or computer science, you can do a lot of mathematical modeling. You can do a lot of prediction how this pandemic will go after 1 year, after 2 year, 3 year. So it's all about your training, and it's all about your question, how you want to see that question and how you want to solve that problem.
So that is the actually should be the aim in the current field of research, or in current for the scope in the current science. Then next question, what is from Subhajit Chakraborty, and he asks, in case of time stress signal, the time difference between two intensity peaks is actually the determining factor, and after the analysis we can get the time constant of the conformational fluctuation or related dynamics. How fast dynamics can we monitor?
Well, this is a good question. So how fast you can monitor dynamics using a single molecule imaging is completely limited by the resolution of the microscope camera. As I told you, the microscope camera is the most expensive part. It's similar like a BMW. The price is similar like a BMW. So the reason people actually invest the money for there is that you can get at least 200 frames per second for any dynamic study. So usually all biological processes are slow. It's a millisecond time scale normally, not biological water dynamics. Biological water dynamics is very fast in picosecond and nanosecond time scale. However, if you want to study any biological interaction, protein-protein interaction or protein conformational change in real time, so that study is completely limited by your camera resolution. Usually all EMCCD have a resolution 1 millisecond to 10 millisecond. But the thing is that you want to go faster. That is, I understand. I want to go also faster, but you also want to get a good resolution, signal to noise ratio. If you go much faster, your signal would be much low, right? Signal to noise ratio would be much low. You almost cannot see any single molecule. So there would be balance between faster, how fast you can go, and then how fast camera can go,
Right.
So let's say camera resolution, if your signal is very bright, you can go up to 1 millisecond time scale. Well, that is a camera, but there is another type of detector. It's called APD. Now, APD is actually single pixel camera. Think about a camera with a single pixel. Now, that APD can actually go much faster. If in a single camera in EMCCD, if you change your region of interest, then you can actually go even 1000 frames per second. So, and you can imagine if your camera is just 1 pixel by 1 pixel, you can go much faster. So when you are actually talking about that, you want to study a dynamics, just not imaging. So it's better to go for APD. So there are many pioneer company, leading company. It's called PicoQuant. You can actually use that PicoQuant single photon avalanche diode for studying dynamics.
So the last actually is not question. I mean, this person, he already asked a question previously that how much it is relevant, the chemistry background or chemistry field, how much it is relevant to that field of your research. And he again commented that actually till now I work only on synthesis of molecules. That's why I asked this question. Okay, I mean, because he is a chemistry student possibly, and he worked on the synthesis, but
Yes.
how his work will be relevant to your field? Can you explain?
See, so I never did a protein synthesis, right? I was a chemist. I was physical chemist. I trained myself to synthesize protein molecule. So when we started this immunology project, we spent 6 months, more than 6 months, 1 year, to make only one protein, which is single chain antibody fragment, SCFB. That will, that will bind on T cell receptor. So that protein actually, making that protein is extremely difficult. So we spend so much time to synthesizing, that protein molecule. Now, again, when we do peptide MSC molecule, there is a peptide around, if it is a class 2 molecule, usually peptide sequence is around 20 amino acid, around 20 amino acid. If it is a class 1, then peptide sequence would be around 8 to 10 amino acid. So we also synthesize peptide, which is a pure chemistry. We use peptide synthesis technique. We purified HPLC. There are many other pipeline to purify peptide. This is also chemistry. Now the thing is that you have to think much bigger beyond that thing, how you are actually applying those things for solving a bigger question. So for example, if you want to synthesize a small molecule, now people will ask today a question, is there any application of that small molecule? What you can do with small molecule? 20 years, maybe 10 years back, when you synthesize a small molecule, maybe complex molecule, you can actually publish a paper. But nowadays you have to show that that molecule have a application. And there you go for biology. So you want to collaborate with biology, or you want to learn biology to do yourself. So I would say you are doing chemistry, excellent, nice, but go beyond that. Apply your technique, apply your knowledge to solve a bigger question.
Yeah, thank you, Dr. Sasmal. I would also like to mention a few things here. That your research, whatever I realized, it's a total interdisciplinary research. If I think about the starting point from physical chemistry, as you mentioned, then you comes to the spectrometry, right? And that is a part of physical chemistry. And then you came to the analysis, instrumental techniques first. And then to use that instrumental technique, you need to use some materials, imaging materials. And especially the few minutes ago, you are giving the answer to the student who is from chemistry background. And also as he's from synthesis background, I will also address for him that the synthesis of several kind of fluorescent dyes, right? Those are nothing but organic molecules, right? And
right.
So who will synthesize the molecule? The chemist have to synthesize that molecule, right? And after they synthesize, once they synthesize, then persons like you or who want to use that one for advanced application, they can use that molecule, right? That kind of dye for as a imaging material, right? And you can use that imaging technique and imaging material, and finally you got some data. But before getting that data, you need data analysis, right? That is a part of computer science, right? And so, and you are applying the whole this kind of combination of this technique for what? To discover some biological process or biological purpose, right? Biology, understanding the biology or human biology or whatever, right? So all in as a I can say it's a package, full package, and it's a kind of interdisciplinary research. So by in that respect, your talk today was really good. In that, how can I say? I would say it is a very insightful talk, and it will be very much helpful for not only our student as well as some persons who are expert in this field and also have an interest on this field, right? They want to continue their research or their study. But last question, what I would like to ask you, this question, the research, whatever you are performing still now, to me it seems like that you are performing to solve some kind of real issues, which the biological field or real world they want. But my question is that how this kind of issue can be utilized for some kind of industrial purpose or to generate some kind of product. That one, if you address very briefly, that will be very good. Because lots of students, nowadays they are more interested for their future career, like after study in this field or after doing research in that field, what will be their future, right? So in that respect, if you address, not only one possible career could be the academic one, but it is not always true for or applicable for all the student, right? So other than that, what kind of field they may choose after doing study in this field, right?
Yeah. So one of the best example after doing PhD from this research field is going to industry. So because everybody will not get opportunity for academic career.
Right.
So the best thing or best suitable the company would be immunology company. You know, there's a company called Genentech in USA in San Francisco. There is a whole team of people who actually develop, you know, immune therapy, different kind of drugs for different kind of disease. You know, it's not about, so the drug company or pharmaceutical company is not just because they just produce a medicine or small molecule. That's the one part. That's a big part, I would say. But there is another part in pharmaceutical industry is that people are working. They're trying to exploit your immune system. You know, they're trying to exploit your immune system to combat different kind of disease. For example, cancer. Many people are trying to combat cancer with PD-1 or PD-L1 inhibitor, which inhibits. So I told you that how cancer cell is intelligent. They actually express PD-L1 molecule or PD-L2 molecule, which is actually present in our normal cell. So when T cell come, they don't understand. They cannot differentiate. T cell cannot differentiate the difference between a normal cell and cancer cell. That's how it invades that. So the question is, the pharmaceutical industry, what they're trying to create small molecule, actually small molecule, maybe a large molecule, a small peptide molecule. They're trying to block that PD-1 molecule specifically on the surface of cancer cell. So once you block, so what will happen? A TCR will come to that cancer cell, and it will think about that this is actually a foreign cell, right? This is a dangerous. So T cell will find this, T cell will form bond with peptide MHC, and then after that it will get activated, and then it will release cytokine to kill that. So there's a whole branch of science and pharmaceutical company. They're trying to solve disease through exploiting your immune system, but they also apply small molecule chemistry, that technique. But their application is different.
Thank you, Dr. Sasmal. I mean, I really enjoyed your whole presentation, and I believe all our attendees also enjoyed a lot. And I don't want to extend more as we don't have much question and comments, but still lots of participants or attendees still with us. But I would like to also thank you to all our attendees for joining this webinar. And before that, I would also one more time like to say to Dr. Sasmal that on behalf of our JISIASR Kolkata family, I would like to request you that whenever you have, you are available, please come and visit our institute. It is available, situated at Kolkata, Short Lake, Sector 5. So, definitely and sure our interaction will be extremely helpful to continue our collaborative research work, not only with me, because I know you from a very long time, because I also forgot to inform our attendees that Dr. Sasmal is one of my very old friend. We were a classmate. We were classmates. So
we are a college buddy.
Yeah, college friend. So surely we can collaborate and we can start again new research in this field. And at the same time, and as our institute also have several renowned faculties, they also will be extremely helpful from your research work and the insights from you. And that's why I would like to request you that please come and visit our institute whenever you are available.
Thank you for your invitation. I'll be very happy to visit your institute.
And also, I would also finally, I like to thank you to all the attendees who have joined this session. And I don't want to say much at this moment. Oh, again, one attendee, he raised hand, Marley Pakase. But I mean, I'm requesting you, plus please don't raise your hand, because only raising hand does not have any meaning here. Because if you have any question or comments, please write. Or otherwise, if you have any question, you can also write in our Facebook Live. I mean, what is going on at the moment, still now is going on at our institute web page, Facebook web page, but it will remain there after closing this session. The Facebook, I mean, recording or whatever the session will be still there on the Facebook page. So you can comment there also, and you can make your question there. So I mean, I will convey your question or transfer that question to Dr. Sasmal later. Okay. So other than that, I cannot see any kind of comments from our audience at the moment. So finally, I would like to, I mean, finish this session, and I would say have a good day and stay happy and stay safe. Thank you very much, Dr.
Have a good day to everyone and stay safe. Bye.
Bye. Thank you.